Good morning. These presentations were extremely well done, and I know John and Karen are just some of the greatest people on the planet in terms of this type of work. What we will do now is take some questions, but before we do that, I want to take the opportunity to introduce Dr. Tanja Gruber, excuse me, from Stanford University, who will be co-moderating this session with me. My name is Barry Mangum. I am a neonatal pediatric clinical pharmacologist. I work on the drug development side and mostly in neonatal medicine. So with that as a backdrop and an introduction, I'd like to open it up to your questions, Q&A. So the first question that we have posted is, what role does pharmacogenomics play in clinical trials designed for neonates?
Do you want me to start taking that one, Barry? Is it okay with you?
I think so, John, because you've done quite a bit of work here with Steve Leader and others. So why don't you take that on?
Yeah, I think it's a great question. I think it's in neonatal pharmacology, there's always this delicate balance between developmental changes based on developmental physiology on the one hand, and then what is there known about the pharmacogenetics. And the question is, which trumps what at what age? And we have learned a lot in all these years that it varies per enzyme on the one hand for the metabolism part, but there's also variation in the mutations that we can have in the receptors. So let me give you two examples. One is pantoprazole. We know it's 2C19. When you're early on in your age as a newborn baby, it's impossible to see whether you're a slow metabolizer or an extensive metabolizer because the developmental changes in 2C19 are more prominent and more important. A few weeks later, that individual with a slow metabolizing capacity can be picked out, and that has been published. On the other hand, so that means that development wins in that respect. 2D6 enzyme, we know after 2 weeks, studies done with tramadol showing that after 2 weeks, you can already see the difference in developmental changes based on the genetic background of the individual. Then going to much more faster things, I think that John Davis showed in a study about neonatal abstinence syndrome, that then you have certain mutations in the mu-opioid receptor or in the catechol-amyltransferase gene. These patients with that mutation have less abstinence syndrome and are having a shorter stay in the hospital. So we are starting to learn something there, but we learned a lot, and that's primarily done by the work in the group from Steve Leader in Kansas City, is that we can't translate the knowledge we have in adults just to the newborn baby. And what we know, and we learned a lot from developmental changes in transporters, we just started to learn there now, and there will be mutations there. But I think as a classical neonatologist, most of the time it will still be development that will trump pharmacogenetics, but there clearly will be exceptions.
John, thank you for that great answer to that question. And I know you have many, many decades of work in this space, and that was good. So the next question is for Karen. How changes in physiology was factored into while performing PVPK models for neonates?
Sorry, can you repeat the question again? How was the physiology for?
How changes in physiology was factored in while performing PVPK model for neonates? How do you factor in the physiology?
Oh, okay. So, so obviously I didn't do a very good job of explaining that during the presentation, but so for each of the parameters, as I mentioned at the beginning of the of my presentation, we have distributions of so we have distributions of organ sizes and also differential equations representing the different processes. When we're talking about neonates, what we do is we essentially start give them an organ size based on their age. and we resample. And so essentially what we're doing is, as they're aging, we're resampling from a different distribution. So their organs are growing. And that is something that we were able to factor into the, say, for example, the ivacaftor lumacaftor simulations that I did. If we didn't account for that time varying physiology for neonates, then we wouldn't have got as good a prediction if we had, because I ran the 2 sets of simulations. And
Great.
also that is done, sorry, with respect to CYP enzymes, organ sizes, most of the physiology that I showed on the actual slide.
Karen, the next 2 questions are to you as well. What is ivermectin drug dosage mentioned in the slide?
So the typical dose for onchocerciasis was what I was referring to there, which is about 150 to 200 micrograms per kilogram. And but most of the original studies had been done at higher doses. So they were the typical doses that were used in the original studies and were used for the model development.
And the next question kind of relates back to the physiology question, and that is that understanding that there are relatively limited info related to pediatric physiology out there in the literature, and most of it's academic. What prominent, what aspects of preterm neonates, especially in the role of bile salts in absorption, can you provide, and how that affects the neonatal physiology and how that will affect pharmacology and PVPK?
So I think, obviously that's a very interesting question, and essentially, I think one of the key things to think about here is how accurate are the predictions. And so I suppose the point I was trying to get across in those slides is that there's a lot of focus on all of the different aspects that could affect the absorption. And in fact, that goes back to the discussion with Hannah Batchelor's presentation yesterday. And there's a lot of focus on those. And the point I was trying to make was that for these BCS class 2 drugs, the bile salt concentration is obviously very important. And even if we don't know precisely what the values are, we can certainly do a sensitivity analysis to have a look at the changes. And the point I was making there was that obviously in a high-fat breast, when the neonate is being breastfed, the solubility may not be an issue. But after the breastfeeding ends and the bile salt concentrations change, then it's interesting to have a look at the effect of the absorption. And as I mentioned during my presentation, this is an area that is being closely looked at, even at the moment in adults, in terms of the bile salt effect. And there are a lot of data showing the levels in neonates, but it's interesting to put them together. And obviously we have to wait for more clinical data in order to be able to verify the impact of the bile salt concentrations. Sorry, that was quite lengthy, but it was quite a loaded question.
No, it is quite a loaded. It's a very complex issue, and PVPK has been around now for I guess at least 2 decades that I know about. And SimSip, the group that you're working with, is extremely knowledgeable in this area, probably the leader in the world. I know that in my space, in the practical aspects of neonatal pharmacology, one of the things we've been going to you guys, and especially Certara, in working with you, is that looking at giving us models that we then can go back and do actual validation of that model with assays from patients. And so I'm thinking that at a high level, what we see is that, and what I recommend for my pharmaceutical companies I work with, is that we do PVPK, and then we validate that PVPK with actual data from the patient population. In this case, neonatal patients. I know you're doing that for some work that I'm doing with you, but others, I don't know. So is that the right way to think about this? Is that we seek out PVPK modeling and simulation, because not everybody can do PVPK modeling, not as well as you guys, because you have the experience. But how is the best way practically for us to approach that when we're dealing with a new chemical entity in a neonatal patient population?
I think, with respect to PVPK, I mean, obviously, as you mentioned, Initially, what we do is we like to develop a model in adults, if possible, and then it gets extrapolated down. And obviously, in terms of applying the model, really what we're trying to do as well is make sure that we've got the disposition, absorption, all of the different components that describe the disposition of the drug well verified in adults. And going back to the ivacaftor lumacaftor example that I showed in my presentation, what was interesting to me is we've talked a lot about reproducibility of PVPK models over the last couple of years. I was able to take that data that had been presented in a publication and used for the combination in cystic fibrosis, run that simulation, and then actually recover what the exposures that were seen in the neonate. And so ideally, what we'd like to do, as you've said, is verify the model, apply it in adults, just because that gives us confidence. And if we have DDI studies that can verify the relative contributions of the components, then we would extrapolate to neonates. But the important thing really about PVPK modeling is it allows you to look at those different processes and to determine what is key, and especially in neonates. Even if we don't have data in neonates, that's the great thing about PVPK.
Absolutely. Dr. Gruber would like to ask the next question, so I'll turn it over to her.
Yeah, Karen, can you comment on the milk composition, because it does change over time, and how that can affect the modeling?
Yeah, so that's a great question. So at the moment, we're just considering exposure in milk, but yes, you're right. What we do need to do in time is, for the first colostrum, the first couple of weeks, when you have that, the different composition that changes after a couple of weeks. Start off the high protein, lower fat, and then switches over, and we need to be able to incorporate that in the simulations going forward. But at the moment, it doesn't. But that does have a significant effect.
Yeah. And another question brought up by an attendee is, when you're considering organ weights, how do you account for variability for neonates that are the same gestational age? For example, a baby who has intrauterine growth retardation, or gestational, whose mother has diabetes, and they may be born with a large liver.
So what we would be able to do is rerun a simulation, or because at the moment what we don't do is we don't obviously have all of those disease or cofactors built in, but we do actually derive a distribution of organ sizes for a specific age based on data that we have from the literature. And so what we would be able to do is rerun a simulation and see what impact change in the liver size has on an individual, and look at that in terms of the simulation.
Yeah.
So in other words, we're mimicking the disease, but not actually creating it within the simulation.
Yeah. And then for biologics development for the young patients less than 2, can you comment on phage organ maturation function approach versus PVPK?
I'm afraid that's something that I can't comment on, but I can certainly follow up on that question afterwards and provide an appropriate response.
Yeah. Okay.
Thanks, Karen. So here's another question for you, Karen. We heard that clearance may be impacted by gestational age, and we see this in the neonatal world. How does PVPK handle the difference in gestational age? Is this a preset parameter in your modeling and simulation?
Sorry, could you repeat that question again?
Yeah, sure. We heard that clearance may be impacted by gestational age. And how does the PVPK handle the differences in gestational age? Is this a preset parameter?
Yeah, so basically it is preset and based on data that we've generated from the literature, and then we're able to change that over time.
Okay, all right.
And have a look at the impact of the change in clearance over time as well. Yeah.
Right.
And that goes back to, my previous point about talking about why it's important to determine, the importance of clearance really versus distribution, because at the end of the day, it may have little impact. And just to mention, obviously, with respect to the ivermectin and moxidectin examples, there's a high biliary efflux component there, and therefore in terms of the metabolic clearance and changes there, that would have a relatively small impact.
Okay.
And therefore, we would have to consider the relative ontogenies.
Right. Dr. Gruber, would you like to pick up the next question?
I think I'm going to say his name wrong, but Ollie's asking how is the correlation and disposition of drugs in preclinical study?